The following is an overview of protein synthesis as a chapter/topic. It briefly covers the steps on how to approach the content, so it mostly matches an entry level of biology. Think 7th grade or maybe 10th grade biology. If you're looking for specific problems to solve or in-depth tips and tricks for more advanced material, there will be other videos coming. Protein synthesis. Oh man, it's like the key to everything in a cell. You want to not get suffering from diabetes? Protein synthesis. You want to grow a pseudopod and leave a hostile environment? Protein synthesis. Proteins are often drawn as these blobs or circles in diagrams, and that's because, like biblical angels, their true forms are too awe-inspiring for mortal minds to comprehend. And this is also where it gets really interesting. So earlier, when we referred to diabetes and, say, pseudopods in amoebas, that's in reference to the fact that almost everything a cell does is in large part due to proteins. You have to remember that proteins are essentially like the automated bots that do everything in the cell, from communication to manual labor to even serving as part of the structure of a cell, like gates or pumps. If you're way into the lore of Transformers, then oh boy, does this serve as a great analogy to proteins. There's a lot of proteins that serve singular functions, but there are in fact some that can transform or even break apart and become something entirely different. And protein synthesis is the origin story. And like all origin stories, it not only tells the story of how they came to be, but it also provides the secret for how they work, including weaknesses and ways to manipulate them. Overall, think of protein synthesis as taking a piece of code, translating it into a sheet of paper, and then folding that paper, origami style, until you get some insane creations. And in most classes, you'll actually be asked to do the decoding. As with the rest of biology, there is no real way to learn this material other than to memorize and practice. Before you get started, you should have already covered organelles, amino acids, proteins. You should have also covered DNA and RNA, at the very least their structures and what their bases pair up like. Keep in mind also that in RNA, thymine is replaced by uracil. On a larger scale, you have to think of protein synthesis in 3 major steps: transcription, translation, and protein folding. In transcription, think like transcripts from a video. You're essentially just getting a copy of what was being said. The data found on DNA is transcribed and copied over to an RNA. In translation, RNA's data is then moved over to a polypeptide chain. And finally, in protein folding, the polypeptide chain is folded until it becomes its full protein 3D structure. Now, when you zoom in, there are 2 major things you have to memorize for each step. The first is location, and the second is the actual process. In transcription, the location takes place in the nucleus. In more advanced classes, you might need to know specifically where in the nucleus, but for all intents and purposes at this level, you just have to remember that since DNA is closely guarded and can't really leave the secure zone, it needs to be in the nucleus. We said earlier that DNA becomes RNA, but you have to be careful because there are usually 3 RNAs types that will be taught to you in this section. The one that we worry about the most is mRNA, which stands for messenger RNA. There's also rRNA and tRNA, which, depending on the depth of the class, will also be covered. As a quick aside, I often geek out when I start talking about rRNA and tRNA because without even going through the full process of protein folding, you have functional parts, including structural parts. These are actually often called structural RNA because they serve a function. They do things in the cell without needing amino acids. And this is where you can really start understanding how viruses work, because viruses often don't have that much inside of them to reproduce with, so they use RNA a lot more effectively like this. Anyway, that's just a quick preview of some more in-depth advanced material we don't need to worry about here. Back to the main topic, we have to ask the question, how does DNA open up? You should have covered DNA replication at this point, which will unfortunately serve to confuse you. The best way to think about this is that DNA replication has to do with reproduction. That's a whole nother ballgame, and here you're talking about more daily functions and manufacturing. So you're not worrying about how DNA keeps itself safe. Rather, you're just thinking about, well, how do I get info out of DNA? So you're just going to have a whole different set of tools. For most intents and purposes, the answer to the question of how does DNA open up is that RNA polymerase does both the unwinding as well as the transcription process itself. Pretty simple. Just answer RNA polymerase. If you're in a more advanced class, keep in mind that there are differences between eukaryotes and prokaryotes. This is a more advanced topic though, so most likely you won't come across needing to answer that. I will cover that in a more advanced module. Now, in the past, I've actually come across some students, as well, unfortunately, as some sources that say it's actually DNA helicase that does the unwinding, but this is invariably not correct. What I'm thinking it is that there might be a misinterpretation that there is the phrase intrinsic helicase activity, and that is what does the unwinding. But you have to be careful and not interpret that as DNA helicase doing the job. Remember, DNA helicase is an actual protein, whereas intrinsic helicase activity is an action. At the end of the day, the answer is RNA polymerase. DNA is not opened up for transcription by DNA helicase. I'll cover this in more detail in future videos, but for now, just remember, RNA polymerase. That's all you need to know. Now moving on to translation. This is part 2 of the protein synthesis. So the first question is, where is it taking place? The most basic answer is the ribosome. But this question can get tricky because it actually depends on what is being asked. In general, if you say the ribosome, that'll work. But they might be asking you where in the cell. Keep in mind, ribosomes are in many places. And in general, you can say it happens in the rough ER. And keep in mind, the rough ER is that way because that's where a lot of ribosomes are embedded, making the surface very rough. But if you remember your lesson on organelles, ribosomes are also free floating. They're just kind of all over the place. So your answer might be the rough ER or the cytoplasm. And for the level of this class, the way to tell the difference between where the protein is being created is to think about what it's meant to do. Remember, the rough ER oftentimes ships things directly to the Golgi apparatus. This is a regular function. And the Golgi apparatus kind of sends things out of the cell, right? So because of that, if the protein is meant as a tool that goes outside of a cell, then it is meant to go to the rough ER. So you will answer rough ER if that's what the point of the whole protein is. If instead it's supposed to do something within the cell, your answer is most likely the cytoplasm. It's not going to be a 100% case if you're really getting into advanced classes. If somebody asks you, is that always true? No, it's not always true, but it's generally true, so you can probably say that as an answer on a more entry-level course. So that answers location. When we talk about the actual process itself, keep in mind again, RNA transfers its data over to a polypeptide chain, and some people will refer to this as the protein. I myself, I don't prefer to call it a protein because it's not functionally one yet. It's kind of like getting a bunch of pieces for furniture from an IKEA box and saying that's a chair. It's not a chair yet. It's all the parts of a chair. It's ready to be made into a chair, but it's not a chair. That's me being very academic. And this is where some classes will ask you, well, what's that ribosome made up of? And then you might have to answer it's made up of ribosomal RNA, rRNA. But almost guaranteed, you're going to be asked about tRNA, which stands for transport RNA. Again, this is RNA that simply folds up very quickly and becomes a structural part of the cell. It is responsible, as indicated by the T in tRNA, for transporting amino acids over to create the polypeptide chain. Each tRNA has a set of 3 bases, which are referred to as an anticodon. This is where it binds to the mRNA to make sure it's delivering the correct amino acid. As a reminder, a codon is a set of 3 bases. This should have been covered in DNA structure, but just in case, I just want to remind you, and that's why when you say anticodon, it should immediately make sense because you already know what a codon is. Now, as amino acids are being introduced, they start connecting to each other, creating a chain. In some advanced classes, they'll ask you what bond connects all the amino acids. It's a peptide bond. And then ask you what that entire chain will be called. As we mentioned earlier, it's called a polypeptide chain. Easy to remember because the word poly means many. Peptide is just a string of amino acids, and usually people will agree that it's between 2 to 50 amino acids. So when you say, hey, there's a lot of them together, many peptides, it's just what polypeptide means. And finally, there's protein folding. Now, luckily, most of you in some of the more entry-level classes won't have to do this part because it can get very dicey and immediately dives into the more advanced parts of protein synthesis. The only things you might be asked to know are the orders of protein structure. Keep in mind that amino acids have these functional groups, and those functional groups are just waiting to interact with something and or not interact with something, which is equally important, and that would immediately make it react to itself. So the term folding is used because it makes sense. If you see a long string of something suddenly bend into crazy shapes because it's reacting to itself, it looks like that string is just folding into a new shape. This is also why I like to use the analogy of origami. You have a flat sheet of paper or maybe just a long string of paper, and you're cutting and bending it and folding it until it becomes a whole new thing. So in all technicality, there are 4 orders of protein structure. But practically, you have to think of it as 3, with the 4th one being a bonus, like you're going A+, you're doing the honors. The first one is known as primary. The primary is just the sequence of amino acids. That's literally what you're translating into. And then there's the secondary order. In that order, you have the smaller local folded structures. In this case, when we say local, we just mean like a part, like a small segment of the polypeptide chain. So in particular, you'll be asked to identify an alpha helix or a beta pleated sheet. I'm not going to go into it in detail here, but there are some classes that will literally just give you a string of amino acids and say, hey, is that an alpha helix or a beta pleated sheet? So watch the advanced video if you need to know how to do that. Like I said earlier, practically, you will finish your protein folding by the tertiary step. So the tertiary step refers to the overall structure. This is actually the whole protein. You're done. Unless you talk about the quaternary structure. The quaternary order of structure is when you have a protein that is not complete unless it has subunits attached to it. Think like you're building a shelf, but you're missing the drawers. And therefore, a combination with other proteins is required. And this is what the quaternary structure talks about, is when you have different tertiary structures all coming together and how they interact in order to become a full piece. Now that ends the memorization aspect of this section. There are actually 2 skills you need to develop when talking about protein synthesis. The first is the skill of transcription. You're going to be asked to look at DNA and then tell them what the RNA sequence will be. For the most part, you're just pairing up the bases, and that's how you're going to get the RNA sequence. Piece of cake. This should be a throwback to DNA replication and DNA structure in general. I like to think that C and G look similar, and that's how I remember that they're paired up. Which, by the way, is the same reason a lot of students mess up their transcription, because they're not careful and their C's and G's look similar, so when they're going back and referencing to what they wrote, they might get confused. So be very careful on the fact that they are similar and therefore paired up. For me, A and T therefore pair as a process of elimination, but I had just learned about what AT-ATs are in Star Wars, so just AT makes sense as a pairing to me. As an introduction, some classes might ask you just to do that, just to pair up the bases and you're done. But in reality, and in most classes that I've seen, they'll ask you to start by looking for the start codon. This is a huge reason a lot of people like the coding analogy, because like in coding, you can't just start typing away code. You need to tell the computer when you're starting your code and when you're ending it. In DNA and transcription, the same thing is happening. You need to tell when you're starting and when you're ending. In particular, you're looking for the codon TAC, which transcribes over to RNA as AUG. And if you do enough practice, these 2 will become extremely familiar to you. TAC, always the part in DNA you're looking for to get started, which then becomes AUG on RNA. Here's a quick tip for people solving these problems. This is a very meticulous type of problem, so you're going to want to make sure you're doing everything you can to keep all your ducks in a row. You'll want to immediately break the sequence into codons, groups of 3. Starting with TAC, draw a line. Then do the next 3, draw another line. Because unless you have an incredibly disciplined eye, you're going to get confused by where you left off. You then continue this process until you find the stop codon. Now there are actually 3 stop codons, so you either have to commit these to memory or make sure you reference the chart, which I'll talk about later. In DNA, you're looking for ATT, ATC, or ACT. When it translates to RNA, you should end up with a UAA, UAG, or UGA. Make sure to check with your teacher though, because sometimes the worksheets or even the teacher are trying to be more realistic. In reality, genes are not that short that you can stuff it onto a problem for students to work on, and so it's not realistic to have a stop codon so soon, which is why they might not include a stop codon. So just check with your teacher that you're not looking for a stop codon. Now me personally, I would take the transcription you wrote, the RNA strand you just created, and immediately break those also into codons and to make sure that there's a line between all of them. That's if you haven't already done so. Now, I've seen some students do the transcription by just pairing all the bases, so you end up with a lot of T's in your RNA strand, and then they go ahead and replace it with U's. Now, this can be helpful for people that prefer to work methodically. You want to do only the pair-ups first, and then properly write the mRNA sequence by switching out the T's with the U's. Personally, I find this technique risky because I might forget to do the step where I replace the U's, and also I see most students not having any trouble just directly switching it to U's. As long as the technique works for you, and you can consistently get correct answers, there's no right way to do this. It's just really important to remember that RNA can't have any T's in it, so make sure you have all U's in it before you submit your answer. And now we move on to translation. The skill involved here is taking that RNA strand you have and converting it into the polypeptide string. Each codon references a single amino acid, and so you either memorize which ones are which, or you reference a chart. I haven't seen a single class that expects their students to memorize it, aside from mine in high school, so you should be safe and just need to reference the chart. The chart, however, has a learning curve, and you definitely need to practice using it. Otherwise, as I've seen a lot of students do, they'll sit there staring at it wondering how they're supposed to get an amino acid out of it. And the reason it's difficult to use is because it's a 3-variable chart. Normally we're only looking at 2 variables, maybe 1 sometimes, but not 3. In the first model, you first look at this side, pick the first letter in your codon, then you go to this side to choose which box it's going to correlate to, and finally within each box you're going to go to this side and figure out using the third letter which inside of box you're going to use. In more recent years, happily there is a more graphically engineered one that looks like this, in which case you move from the inner circles to the outer circles to figure out which amino acid you are. By the way, side tangent here, if you're interested in the way one chart works way better than another, you're going to want to look into the interesting blend of graphic design and science. And for those of you that play video games, that chart might actually look similar to some interfaces that you use in your games. And that's because graphic design is all about how do you place things and in what way in order to make it as accessible as possible. And that's it for the skill sections of protein synthesis. If you cover the topic more in depth, you might start answering some of the questions you might be wondering when you're covering this. As a quick sneak peek into that realm, keep in mind that a free floating DNA or RNA that's opened up and ready to be replicated or transcribed is extremely dangerous. This is why there's a lot of proteins and a lot of regulation put in place in each cell to make sure that doesn't happen. Keep in mind, something like a DNA just sitting around ready to be replicated, not being closed up and guarded, can cause things like cancer. That's how serious, that's how bad this can be. In fact, a lot of diseases are basically because you have random floating DNA or RNA. That's essentially how viruses do what they do. So if you're interested in learning what kind of regulations and how your cell stops DNA and RNA from just randomly doing whatever they're supposed to be doing, this is when you want to go into the more advanced course. Now, I have a particular bias towards protein synthesis because my major was in essentially genetics, which, if you're interested in that topic, protein synthesis becomes your bread and butter. I can happily report that within the world of genetics, you don't usually have to transcribe and translate like you would in a classroom. That job is normally done by a machine or software. But you have to be good at doing the job itself because your ability to analyze as well as double check and troubleshoot is important. The machines are not infallible, and it doesn't necessarily know what it's looking at, so it's up to you to be able to understand what that information is. I spent one semester doing nothing but constantly sequencing some yeast cells because we were hunting for a particular growth gene, and in that process, I practically memorized the entire chart of codons to proteins all over again. I had forgotten it after high school, but that class, oh my god. And protein folding is almost an entire industry in and of itself. I often compare proteins to nanomachines, and that's because that's essentially what they are. And this is what I was referencing to when I said if you understand the origin story, you understand the weaknesses and the manipulation. If you know how a protein folds, you can go in and mess up, or, in our case, fix the DNA and know how to make it work either back to normal or even better. And amazingly, there's actually an application, a game in fact, called Fold It. You might want to look into that if you're interested in the world of protein folding. The whole idea behind Fold It are two main concepts, thin slicing and gamification. In terms of psychology, thin slicing is the concept of somehow learning the heuristics or the methods behind something just through a very, very small set of experiences. For example, there were pilots reported as somehow knowing exactly what size bullet holes and where it can be found on their planes after they do a mission, and when they come back and then engineers ask them, well, where do you think you were hit? They were almost incredibly accurate with where they thought just because of the way the plane moved. So if you want to take advantage of thin slicing in something like protein folding, one way is to turn it into a video game. And this is where gamification comes into play. Gamification is simply trying to figure out how to psychologically get you to do something or behave a certain way. Your teachers do this all the time. They assign point values to certain behavior or certain things they want you to do more of, and they give you extra credit when you do even more than that. So Fold It rewards players by making them do actions that would actually do great protein folding techniques. And so even if you didn't really understand what hydrogen bonds were, why proteins have to fold this way or another, you start becoming an expert in protein folding. That's where the thin slicing comes into play. So it was created by the University of Washington as an experimental research project to see if combining those two concepts would be able to support the research of protein folding. And within months of its release, there were already reports and articles written about some of the really effective results of players being on Fold It. I remember hearing a few years later that there was a 15-year-old problem that was unsolved by the scientific community, and Fold It was credited as being the major reason why they could solve it eventually. And if my memory serves, I can't actually find the article right now, there was actually an NPR segment where they talked about a high school student who just got bored and became one of the top players on Fold It and really contributed some of these. Fold It's not for everybody because it is, after all, protein folding turned into a game. How much of a game can you really make it, right? It's not like it's got swords or supermodel looking characters or great story or music. But as somebody who is very convinced that gamification is really, really needed for education, this was a huge success in my mind. And that's the basics of protein synthesis. And if you have any questions, please drop a comment and I will try to get back to it as soon as I can.